Hello and welcome to another pre-lab video here in Chem 106. So the lab that we are discussing today is the equilibrium constant for the esterification reaction experiment. And basically the reaction itself, we are taking acetic acid and reacting it with propanol. And this is normal propanol, so 1 propanol. And those will react with each other to make water and this ester compound. The ester compound is known as propylacetate. But what we are doing with that is we are going to set up a synthesis to create this reaction, and we're going to monitor the reaction through a titration by measuring how much acetic acid is present at the beginning of the reaction through a titration, and then how much acetic acid is present at the end of the reaction through a different titration. And so that'll allow us to calculate the initial concentration of acetate, the final concentration of acetate, and be able to figure out what the other concentrations are just by extension of some simple stoichiometry. So the question is, how do we go about doing this? And so if we look at the procedure, the first part of the procedure is all about setting up something called a reflux apparatus. Now, in the lab manual itself, we give you this teeny tiny picture of what a reflux apparatus looks like. But I think we might be better served to take a look at a little bit more detailed pictures that are actually pictures instead of diagrams and illustrations. So here we go. So in the experiment itself, you are going to be using a round bottom flask. That's what this one is right here. And you can see they're pouring the solution into that. That's what you're going to do. So the pre-lab calculations have you figure out just how much of the propanol, just how much of the acetic acid that you need to measure out. And so you're going to measure those things, and then you're going to pour them into this round bottom flask. That is your reaction vessel. Now, you are going to attach to that vessel, to that round bottom flask, a condenser. The condenser is a water jacketed piece of glassware. And that water jacketed piece of glassware has 2 ports where you are going to connect this to the water line at your station. And so the water input line goes here on the bottom. The water travels up the tube and then comes out the top. And then this tube goes into your sink drain. Now, notice a couple of other things here in this illustration. Notice that this particular apparatus is clamped twice to this ring stand that is immediately behind it. There is a clamp here on the round bottom flask itself, and there is a clamp here on the condenser, the water jacket. And notice that they are different kinds of clamps. This is a utility clamp down here. This is a 3-fingered clamp here. The main reason for the difference is this water jacket on the condenser is usually wider than where most of your utility clamps can go. So you have to use a 3-fingered clamp instead. Now, one thing that we will be doing that is different is we will not be using a heating mantle. We will be heating this directly on a hot plate. Now, to help with the prevention of what is called bump is when big bubbles form inside of the solution and they cause the solution to splash around all over the place. To prevent that, we are going to be using some boiling stones. And the purpose of the boiling stones is that they will react or act as a place for bubbles to form so that we get nice small bubbles that are not going to go wildly out of control. So this apparatus will be set up on a hot plate. And the nice thing about the hot plate is you've got relatively easy control for heat. So if things start to go crazy, you can turn the hot plate down. If you want things to start cooling down, you can take the hot plate out completely. And because it is still clamped to the ring stand, you should be able to very easily maintain everything that you need to. Shouldn't fall down or have to worry about anything falling and breaking. And so this is the apparatus that you're going to be doing. Now, to set up the reaction, that is what you're going to do. You're going to do, like I said, a series of titrations. So the first titration you do is going to be off of this initial set of combination. So you have your acetic acid and your propanol. You're going to take a 1 milliliter sample of that solution, titrate it with some standardized sodium hydroxide, and figure out what the concentration of the acetic acid is. If you did the prelab correctly, you know that you have equimolar amounts of acetic acid and propanol. So whatever the concentration of the acetic acid is, that will also be the concentration of the propanol. So you do the initial concentration, the initial titration at T equals 0. While you're doing that titration, your other partner finishes the setup by getting a drop of sulfuric acid, or I'm sorry, 8 drops of sulfuric acid, and adds them to the round bottom flask, gets it set up on the hot plate, starts the refluxing reaction. And what does that reaction look like? Well, let's scan down a little bit further here. What you will see in the refluxer is you're going to see something along the lines of what is called a reflux ring. So these on the left here are what happened when you do water in this refluxing column. You see it gets kind of blurry here because you've got all of these water droplets that are condensing and kind of clouding up. What you're going to see instead is something more along the lines of what you see right here. You're going to see some small droplets forming just as the gas is traveling up the tube. It gets to the cooler air and water in the condensing column. It starts to bead, and eventually those beads get big enough, gravity drops them back down into the reaction vessel. But you should see some sort of a cloud ring that forms inside of the vessel itself. And that is a sign, that is called the reflux ring. That is a sign that you're on the right track. What you're looking for is you want that reflux ring to get up above this first notch. So somewhere between 1/3 and half of the way up the condensing column. If it goes too high, if it starts going above that, that's a sign that you're actually heating too rapidly. You need to downgrade the heat, lower the heat. If it starts to really go bad, take the heat off completely and let the whole thing cool down. So you're going to reflux that whole thing for an hour. So 1 hour of boiling. At the end of the hour, you're going to take it off the heat, let it cool down. So let it cool down enough so that you can actually handle it. Once you are in that position where you can handle it, I'll have ice baths ready. You can dunk it in the ice bath. Just make sure that the water doesn't go into the flask, because since we're interested in the amount of water that's present, any water that gets introduced into the flask that's not supposed to be there is going to impact the position of the equilibrium and your ultimate results. So at the hour mark, we cool everything down. We take out another milliliter of the sample to titrate it. And then we dry off the whole thing, put it back on the refluxing column, and get it to heat again. And the reason that we're getting it to heat again is because at the 80-minute mark, we are going to do this whole process again, where we take it off, we cool it down, we take a sample, we titrate it. Now, why do we go through all of that? Well, hopefully, at the 80-minute mark, we will find that the position of the equilibrium is effectively the same as at the 60-minute mark, which means that the reaction has reached equilibrium, and we can go through the process of actually calculating the equilibrium constant. If my titrations at 60 minutes and 80 minutes are vastly different, meaning that the reaction has continued to proceed, then we would need to try to repeat this again, heat it up again until the 100-minute mark, and hopefully that would be enough. So what are you doing while this refluxing is occurring? So in stage 1 here, during that first hour, there are 2 things that you need to do. One of those things is you need to do the initial titration and figure out what the starting concentrations of the acetic acid and the propanol are. The second thing that you need to be doing is calibrating your dropper. One of the things that you did after you did this initial titration, one of the things that you did was you added concentrated sulfuric acid to the solution. Well, as you know, concentrated sulfuric acid is an acid, just like acetic acid. And the titration that you're doing is going to have extra acid in it because of that. So you need to calculate a correction for how much of your titration that you do at 80 minutes and at 60 minutes, how much of that is related to the sulfuric acid that you added. Now, why add sulfuric acid in the first place? Well, it's a catalyst. The purpose of the sulfuric acid is basically to catalyze this refluxing synthesis reaction and make it so that it goes faster. Like I said, we're trying to get this to take place over the course of about 80 minutes as opposed to about 3-4 hours. So the catalysis is really helping with that. That's why we're using the sulfuric acid. So that is ultimately what you are doing in this experiment. In the end, what are you calculating? What are you collecting? Well, you need your initial volumes. You need your calibration data. And then what this allows you to do is, this is your titration data table for the titrations that you did. And this is the process that you follow to do a correction factor. At the end of this, you will calculate a value in milliliters, and that is the number of milliliters that you subtract from this volume to get the corrected volume. Now, for the initial titration, the corrected volume is going to be exactly the same as the original volume. And the reason it's exactly the same is because you should have taken out the 1 milliliter before adding the extra acid to it. So it doesn't need to be anything more than what it already is. Based on your corrected volumes here, you can calculate your acetic acid concentrations, you can calculate your propyl concentrations. And again, since you measured out equimolar amounts, they are going to have the same concentration. From here, you can start to fill out your ice tables. So your initial concentrations of acetic acid and propyl alcohol would go here. Initially, you would be starting with 0 water and 0 propyl acetate. Your final reading would give you the equilibrium concentrations, which would allow you to calculate the change and therefore the equilibrium concentrations of the products. And you can then use that to figure out the value of the constant. So the experiment itself is very straightforward, but the trick to it is you are doing some things that you've never done before. You're using some equipment that you've never used before. And by the way, that some of that equipment is on the more expensive side because, I mean, it's that round bottom flask. It requires a little bit of money to make something that is that hard and that temperature steadfast. So do be careful with all of the equipment for that very reason. But in terms of the calculations are very straightforward. They're not terribly difficult to do. It's just a time thing. The reaction itself takes at least 80 minutes to run. And if you're